Hello, everyone. I'm Anas Al Ubaidi, and this is the eighth lecture or eighth session within Chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. As the chapter is about conceptual and theoretical monitoring, while the topic for this session is about monitoring of drug observation and drug measurement interaction trials. Introduction. We'll start with the definition. An introduction. As drug observation interaction trials focus on the interplay between medicinal interventions and clinical observation methods, investigating how observational biases or diagnostic techniques impact drug efficacy and safety outcomes. While drug measurement interaction trials analyze how different measurement tools or methodologies, as for instance laboratories assays, imaging systems, influence the evaluation of a drug's pharmacodynamics and pharmacokinetics. Significance ensures accurate drug efficacy and safety data by mitigating biases introduced through observational techniques or measurement inaccuracies, enhances the reliability of clinical trial results, aiding regulatory approval processes and patient safety, identifies discrepancies in results stemming from differences in observation methods, as for instance, for example, subjective clinician reporting, or analytical tools, as for instance, manual versus automated lab assays. As for example, during a study on antihypertensive drugs, varying blood pressure cuff calibrations led to inconsistencies in efficacy results. Trials must standardize equipment to ensure reliable data collection. Key points. We'll start with the drug observation interaction. Again, the definition for the drug observation interaction as drug observation interactions refers to how biases, errors, or variability in observational methods affect the assessment of drug outcomes. These may include subjective clinician evaluations, variability in diagnostic reporting, or patient self reported data. Key focus area here we have the subjective clinician reporting. Clinical observations are often subjective, relying on the judgment and expertise of healthcare professionals. While valuable, this subjectivity introduces variability. And here is a summarized diagram summarizing the impact of observational biases on drug outcomes. As here we have subjective clinician reporting and patient reported outcomes, and the share area, common shared area is observational bias. Continuing with the key focus area. But first, we have two examples about the subjective clinician reporting. As example one, a rheumatoid arthritis, or RA for short, trial evaluating joint swelling severity showed discrepancies between different clinicians using visual grading without standardized protocols. Example two, a depression trial relied on clinicians' emotional tone assessments, leading to inconsistent therapy efficacy grading. We have another key focus area, which is the patient-reported outcomes, as patients often report symptoms such as pain, fatigue, or discomfort, which are inherently subjective. Variability in language, pain thresholds, or memory recall can bias results. As for example, a migraine trial relying solely on patient-reported headache duration found wide discrepancies in self-reported data due to differences in pain tolerance and recall ability. Behavioral observations. Observational reporting on patient behaviors, such as activity levels or compliance, requires consistency to prevent bias. As for example, a weight loss drug trial using step centers found differences in monitored activity levels due to patient variability in how wearables were used or interpreted. While here we have now the drug measurement interaction. As a definition, the drug measurement interactions analyze how the tools, devices, and methodologies used to capture clinical data influence trial outcomes. These interactions highlight potential errors arising from inconsistencies or limitations in measurement practices. The key focus areas here we have device calibration. Proper calibration ensures that measurements or measurement tools provide accurate and consistent data. Variable calibration can skew results, especially in multi center trials. As, for example, blood glucose monitors in a diabetes trial demonstrated output variability due to differences in calibration intervals across clinical sites. Standardized imaging protocols, imaging techniques such as MRI, PET, or ultrasound are indispensable tools in clinical trials. Variation in operator handling or imaging settings affect result reliability, as, for example, cancer imaging using different contrast agents or MRI parameters at participating sites led to inconsistent tumor response evaluations. Automated versus manual methods. Automated systems often minimize observer bias or errors, but manual evaluations retain some diagnostic nuances. Balancing these methods is necessary for trial success, as, for example, manual histopathological grading of biopsy slides varied between pathologists, while automated scoring tools offered more consistent grading for clinical endpoints. Multi site measurement consistency. Larger scale, multi center trials often struggle with standardizing measurements across sites, demanding or rigorous standardization. As, for example, a lipid lowering drug trial faced challenges when different labs use varying assay kits, impacting plasma cholesterol readings. Steps to effective monitoring. Here, first, we have a summarized diagram on ensuring consistency in clinical trials. We have start from left to right planning, execution, data analysis, and then reporting. So, step one planning clearly defined protocols for all observation and measurement methods are essential. These protocols must address calibration, site training, and consistency. As, for example, a brain tumor trial implemented uniform MRI imaging protocols to prevent site discrepancies in lesion measurements. Step two execution. Clinicians and trial staff should be trained to apply standardized observational and measurement techniques consistency. Example, rheumatologists in an arthritis trial received training on using ultrasound imaging instead of subjective visual grading to assess joint swelling. Step three data analysis. Analyze discrepancies between sites, devices, or observers to adjust variability. Advanced statistical approaches can identify and reconcile data inconsistencies. As, for example, comparing pre adjusted patient reported scales with objective electroencephalogram (EEG) readings in a sleep trial improved efficacy conclusions. Step 4 reporting addressed the impact of observation or measurement methodologies on overall trial results and final reports, highlight standardization as a key factor for reliability. As, for example, a cardiology drug trial highlighted how pre calibrated echocardiography equipment reduced variability in cardiac output measurements. Now we have tools and technologies of monitoring. As here, we have a summarized diagram on enhancing clinical trials with technology. As we have here, digital reporting platforms, automated laboratory equipment, standardized imaging software, wearable technology, and also quality control systems. So we'll start here with the digital reporting platforms, equip trials with centralized apps to capture clinician observations or patient-reported outcomes. As, for example, a centralized mobile app streamlined clinician input on system scoring during a pain relief trial. We have automated lab or laboratory equipment, tools such as flow cytometers, mass spectrometry systems, and automated immunoassay platforms improve consistency in trial laboratory data. Example, automated plasma glucose measurement eliminated technician-based assay errors in a diabetes trial. Standardized imaging software, tools that enforce standardization for imaging data ensured greater accuracy across trial sites. As for example, RESIST software analyzed lesion seizures uniformly across multi-center cancer trials. Wearables provide real-time patient data, including heart rate, activity level, or sleep quality, reducing observation gaps. As for example, an insomnia trial linked actigraphy-based data with clinical observations to offer a holistic view of patient sleep patterns. We have quality control QC systems. Automated QC systems ensure routine recalibration of measurement devices throughout a trial's lifespan. As for example, hematology devices used in thrombosis trials underwent QC quality control checks biweekly to detect measurement drift. Summary tables now. Next, we'll revise what we have covered through this lecture through some tables. We have a table on key components of monitoring: component description, objective, example, outcome, components, patient recruitment, data collection, regulatory compliance, bias mitigation, consistency assurance. Then we have another table on the steps to effective monitoring. As a step, objective, key actions, example, outcome, steps, we have planning, execution, data analysis, reporting, and risk mitigation. Now we have table on tools and techniques for monitoring. Tool, technique, function, features used, example, advantages. As tools, techniques, we have digital observation apps, automated assays, imaging protocols, data harmonization tools, quality control systems. Table on examples of successful monitoring: scenario, challenge, solution, impact. Scenarios, we have standardizing observations, laboratory assay variability, real world monitoring errors, imaging variability, outdated measurement tools. We have table on applications of key techniques, technique, application, example, insights gained. Techniques, we have digital observational apps, automated lab assays, standardized imaging, data harmonization tools, quality control systems. Application table. As here we have scenarios for each scenario: key principle, applied key actions, timeline, what to watch out for. Scenarios, we have managing variability in blood pressure measurement tools during antihypertensive drug trials, comparing subjective clinical visual assessments to objective imaging in arthritis trials, improving recruitment for diabetes trials with diversely calibrated glucose monitoring systems, standardizing MRI protocols for tumor response measurement in oncology drug trials, mitigating discrepancies in EGFR results in renal drug studies using varied measurement methods, tracking hemoglobin A1C consistency in diabetes management drug trials, resolving subjective endoscopy based assessments of gut health in gastroenterology trials, evaluating patient reported pain levels using mobile tracking apps in chronic pain drug trials, ensuring regulatory approval for novel glucose measuring device integrated in diabetes trials, enhancing PET MRI imaging consistency in cancer tracer studies, resolving clinician bias in subjective pain reporting for arthritis treatments, addressing variability in manual grading scales for dermatology trials, improving calibration of ambulatory devices for cardiac drug evaluations. Next, we will revise what we have covered through this lecture through some MCQs or multiple choice questions. Question 1. What is the primary focus of drug observation interaction trials? Is it A, evaluating biases introduced by clinical observation methods, B, analyzing the impact of laboratory assays on drug measurements, C, comparing patient responses across different clinical trials, or D, enhancing resource allocation for recruitment? So the primary focus of drug observation interaction trials. So A, the answer would be evaluating biases, errors introduced by clinical observation methods, as drug observation interaction trials aim to study how clinical observation methods such as subjective clinician reporting impact the evaluation of a drug's efficacy safety and safety. Question 2. Which strategy helps standardize imaging protocols during trials? A. Using clinician reported assessments only. B. Weekly calibration of imaging devices. C. Adapting fixed protocols like RECIST for tumor measurement. Or D. Allowing flexibility imaging interpretation across sites. So the answer is C. Adapting fixed protocols like RECIST for tumor measurement. Standardized imaging protocols such as RECIST are crucial for ensuring uniformity in therapeutic response evaluation, reducing site to site variability in clinical trials. Question 3. What role does quality control QC perform in drug measurement trials? A. Speeding up the recruitment process. B. Providing personalized training to recruitment staff. C. Identifying and eliminating errors, bias through regular calibrations and assessments. D. Scaling down trial costs by limiting devices used. As here from the four choices, the only one applies to QC quality control would be C, identifying and eliminating errors or bias through regular calibrations and assessments, as QC systems involve routine calibration and blind reassessments to remove errors in measurement devices and ensure the accuracy of collected data uh, during trials. Question 4. What is data standardization critical in why is data standardization critical in clinical trials? A. It speeds up trial completion rates. B. It harmonizes data collected from varying observation and measurement tools. C. It reduces patient dropouts during the middle of trials. Or D, it ensures trial size can shift protocols dynamically. So it is B, as it harmonizes data collected from varying observation and measurement tools. So B would be the answer. Data standardization combines results from different tools and methodologies into a consistent format, ensuring reliability across trials and improving data interpretation. Question 5. How can discrepancies in clinician reported data to reduce to be reduced in trials? So how can discrepancies in clinician reported data be reduced in trials? A. Using automated observation tools and standardized protocols. B. Employing multiple clinicians for each observation. C. Increasing the frequency of manual assessments. Or D. Limiting trials to one clinical site. So the answer is A, as using automated observation tools and standardized protocols. A would be the answer, as automated tools and standardized protocols minimize human error, ensuring that observational biases or subjectivity in clinician reported data are reduced for consistent trial outcomes. And here we come to end of this lecture. Thank you very much and goodbye.